In this chapter, we're looking at how cells make energy, and there's some really cool ways that it is done. So stay tuned here. This is going to be a fun one. Okay, a little bit of semantics here before we get started. This is called aerobic respiration. Respiration is a general word that means anytime there's an exchange of gases happening. So, like for example, we would use this on a more macroscopic level to talk about breathing. You know, if you're respiring, it means you're breathing. In physiology, we would talk about external respiration. That would be the exchange of gases between your lungs and the blood, or you can talk about internal respiration, the exchange of gases between your blood and the rest of your cells. And when I say aerobic respiration, we're talking generally about cellular respiration. And cellular respiration is the exchange of gases that's happening intracellularly, just inside, for example, one cell. Now, this can be aerobic or it can be anaerobic. Aerobic means it requires oxygen. Anaerobic means it does not. There are some, single-celled organisms, some bacteria and archaea that are anaerobes, meaning they don't require oxygen. They would require something more like sulfur or nitrates or sulfites or something. But most eukaryotic cells, and the eukaryotic cells that we have, would be aerobic respiring cells. So we'll spend most of our time here talking about this particular process. Okay, here's a general equation for how this shakes out. You have a molecule of sugar, generally. You can start with other types of molecules like fats or proteins, but generally the end result is the same. They get converted to a sugar. And we take in oxygen, and together they undergo a series of reactions, and some of the products that are made, we have a byproduct, a gas, carbon dioxide. 6 molecules of it are made. We have some water that is produced as a byproduct, 6 molecules of it, and then we have energy that's produced. Now, what is the energy that is most commonly used by the cell? That is an important molecule known as adenosine triphosphate, or ATP. And here is a schematic of it. What I want you to notice here is we have this part of the molecule is adenosine, and onto it are 3 phosphate groups. Each one of these here this is showing you an atom of phosphorus bound to oxygens. These are all phosphate groups. Now, what's you should remember is that oxygen is very electronegative, so anytime you have a lot of oxygens that are attaching to an atom, generally it's going to lead to, they're going to pull and hog the electrons more frequently, and so it will lead to a partial negative charge. And in above and beyond that, we actually have a full negative charge on this molecule. So think about this then. You have a phosphate group here that's got a negative charge on it. Then to add another phosphate group on, you have to get a negative charged molecule onto another negatively charged molecule, and that just doesn't happen naturally. Negative charges repel negative charges, and positive charges repel positive charges. So the cell actually has to force this second phosphate onto the group to make ADP, or adenosine diphosphate. So then comes the last phosphate. Here we have the 3rd phosphate. And to add a phosphate onto adenosine diphosphate is really difficult. You have to invest a lot of energy to make that happen, because you already have 2 negative charges here that are begrudgingly here. And now you have to try and fit a group that's got 2 negative charges against those 2 negative charges. They do not want to be close to each other, not one bit. Okay. So this is called a high energy bond, because it takes a lot of energy to get that 3rd phosphate onto ADP. But if it can be accomplished, then this bond stores a lot of energy, and as soon as this phosphate can get off, it wants to get off as quick as it possibly can. So, and when it does, it releases a lot of the energy that was originally put into making this bond occur. So this molecule then sort of becomes, we call it the energy currency of the cell, and it's similar to any other currency. Think about a currency. A currency in and of itself is useless, right? The paper dollars I have right now in my wallet are mostly useless. Like I might be able to use them to start a fire if I needed to, but in and of themselves, they hold no value, but they are a placeholder for things that are much more valuable to me. And so I can use the energy stored in those dollars. Like I get those dollars from the energy I exert in teaching, and then I get paid a salary. And so for the energy that I exert in a useful thing such as teaching, I'm giving a useless entity that holds the value of my efforts. And then I can, because we all agree that that thing is valuable, I can take that to the store, I can give it to a storekeeper, and I can use it to buy something that's of value to me. So the energy that I put into teaching, out of it, I can get something very useful to me, but in the process, in the exchange, we have to use a medium that is, mostly useless. So that is what ATP is. The cell is not using ATP. It's not like stacking ATP up to make cell organelles or molecules or things. The molecule itself, ATP, is not really used up. But it does get used as a placeholder for energy that can be sort of the transaction of energy through the cell. So ATP is made, it stores energy, it goes all to different parts of the cell, and it breaks apart and releases that energy to then do things that are valuable for the cell. Some of the things that ATP is most commonly used for, it's used a lot for muscle contraction. You spend a lot of ATP contracting your muscles. You spend a lot of ATP pumping things from a low concentration to a high concentration. That would be more at the microscopic level, using ion pumps to pump ions out of the cell or into the cell, up their concentration gradient. And the cell would spend ATP to do things like build macromolecules. Okay, any of that type of building or pumping, that all requires the energy stored in ATP. So really valuable molecule. How is it made? How do we actually get that third phosphate onto ADP? This is a super cool story that I've just totally fallen in love with, and I hope to instill some of that passion to you as well. Okay, what's really cool about this process, how we make ATP, is that it utilizes a sort of a universal principle that we have also discovered and utilized in our technological advancements. So what you're seeing here is a water wheel. And long before the internal combustion engine and the steam engine, during the medieval times, the ancient times, we discovered that we could use energy stored in water to drive mechanical machines that could then be used to build things. So how this works is that water has a lot of energy stored in it. And just to imagine how this would be the case, think about like if I gave you 2 5-gallon buckets of water and you put it in a creek and you had to carry it up like 1,000-foot mountain. It would take a heinous amount of energy to carry that heavy water all the way up the mountain. But if you did, you could actually regain that energy when the water flows back down. And that's how a water wheel works. You have water that's been pumped up somewhere, or it's at a higher elevation. Usually it's because they've dammed up a creek or something. And so you have water that's sitting at a higher elevation than the outlet. And so gravity causes that water to flow down, and that water is heavy. And so as all of this heavy water is flowing down, it can pull on something like a water wheel and cause it to spin. And then that could be hooked up to a drive shaft and used to operate machines. Well, this is in essence what the cell is doing, except instead of using water, it uses a nanoscopic particle called a proton. So take a look at this. This is a picture, this is an illustration here of what's known as the ATP synthase. And these are proteins in your cells that, as the name suggests, make ATP. What you should notice about an ATP synthase is that there is an accumulation of a particle on one side of it and not so much on the other side. So there's a concentration gradient here. You see that all these particles up here, there's a lot of them, and there's not so many down here. That is sort of like akin to the water wheel, right? That is, this would be like, think about if this was a water wheel, think about this being like the water stored at a higher elevation and being used to drive a mechanical process. So this protein has a rotor in it, and as that rotor spins, it actually forces an ADP to attach to a phosphate group. And what you're seeing in this illustration is you have these blue molecules coming in. Those are the ADPs. They are adenosine diphosphate. They only have 2 phosphates on them. And then you'll see this little other particle come whirling in here. That is a single inorganic phosphate, and that will attach to the ADP through a high energy bond to form ATP. So this cellular machine here is forcing those two together. It's forcing the phosphate and the ADP together, even though they don't want to have anything to do with one another. And in it, it will store an immense amount of energy that can be used to go do work. Now, how this specifically operates, what at the nano level is causing the ADP and the P to come together, I'm not entirely sure. There's a lot of really interesting biophysics there, I think. If you want to learn more about it, do a deep dive and tell me about it. It might be ripe for a bonus challenge worth some bonus points. But what I want to show you, though, is that the thing that's driving the rotor so that all of this can happen, okay, is the flowing of protons through these channels. Think about it being like a water wheel. As it flows through, it causes the turbine to spin, and that's what powers the cellular machine. Now, we call these protons, we also call them hydrogen ions because they actually come from hydrogen. Hydrogen is a very simple atom. It has 1 proton and 1 electron. Okay. Now remember, if you have an ion of an atom, it means it's charged. And so the hydrogen cation would be a proton. And we would say it's H+. So all that to say, when you're looking at diagrams of this, these might be labeled as protons, like P, or they may be labeled as H+. They are one and the same thing. They are simply a proton. So if we can accumulate a lot of these positively charged protons here, they don't want to be next to one another. And so it takes a lot of energy to get these protons to accumulate here. But if we can establish what's known as the proton gradient, then the energy stored in it can be used to drive the ATP synthase to make ATP. And then that is literally, think of it being like the power generator that, or like the water turbine, you know, think about Hoover Dam, the hydroelectric plant, whatever you can think of, it's the energy generator of your cell and thereby the energy generator of all of life. It's a really cool machine. And I could just stare at it all day. Okay, now these are found in prokaryotic cells. They're also found in eukaryotic cells. They're found in different places depending on the cell. If we're talking about a prokaryotic cell, this would just be found in the outer plasma membrane. So that's what you're seeing here, that you're seeing the phospholipid bilayer, and a part of it is anchored into that membrane so that it's, you know, it has some stability to it. It's not just floating loosely in the cell somewhere. Now, in a eukaryotic cell, these are most commonly found in an organelle known as the mitochondrion. So here's an illustration of a mitochondrion. And mitochondria have 2 membranes to them. They have an outer membrane and they have an inner membrane. And the space between those is called the intermembrane space. Okay, that is what you're seeing right here. That is the place where protons accumulate. That is where the proton gradient is established. So in an illustration such as this, you could say anywhere there's this lighter green color, this will probably be a place where there's high concentration of protons. And anywhere that's in this pink spot, this is called the matrix of the mitochondrion. It's just the innermost part. And probably in the matrix there would be a low concentration of hydrogen ions. And so you then can have a concentration gradient, and the ATP synthase proteins would be found all along this inner membrane. Okay, and so anywhere that they are, they act sort of like a doorway to allow the hydrogen ions to flow through and generate ATP. Now, the question that remains then is, how do we establish the proton gradient? How do we get, this is a pretty simple process in and of itself. By the way, this is called chemiosmosis because it's like an osmosis of chemicals in a way. I won't ask you about that on the exam. That's more of a term, but chemiosmosis is what we call it when hydrogen ions flow through this. But how do we establish the proton gradient in the first place? Once it is established, then it's pretty simple. They just flow through and churn the ATP synthase to make ATP. But how do we get them here in the first place? How do you get a bunch of similarly charged things to get close to one another, against their will? That is what involves a pretty complicated, tangled labyrinth of processes, and that is going to be where we spend the rest of our lecture. And how does this happen? Okay, it all begins with a molecule called glucose. Glucose is a monosaccharide. It's a sugar, and it has the formula C6H12O6. Now, essentially, this molecule gets broken down, and the carbons and the oxygens, they form CO2, and you breathe it out. So think about this for a second. I always tell my students, a majority of the molecules that you eat when you ate breakfast this morning, if you ate breakfast, think about the donuts or the cereal or whatever, what happens ultimately to a lion's share of those molecules is you breathe it out. Isn't that wild? You are literally breathing out your breakfast right now. I just think that's amazing. Okay, so carbon is sort of like a scaffold atom. It usually doesn't get used to do much other than hold the molecule together, so we get rid of that. Oxygen, we get rid of that. Now, what's highly valuable in terms of energy production is the hydrogen atoms. And in fact, if you want to know how much energy a molecule can provide, you count up the hydrogens. Things like fats have tons of hydrogen on them. And so a fat molecule is going to hold a lot more calories on it than something like a sugar molecule. In essence, what happens is pretty simple. The hydrogen atom is split into its parts. So it's split into a proton, split into an electron, and sort of like absence makes the heart grow fonder, right? These are separated for a time, and it's sort of a cool story. As they are separated, they do what is necessary to provide for the cell. The electrons are used to establish a proton gradient, and the proton gradient then is what's used to make ATP. Cool story. Now, this is going to be pretty intensive. How this actually works. If we look specifically at the different processes involved that making that happen. But in essence, it's fairly simple. The hydrogens are split into their parts, and they are used to establish the proton gradient, all for the purpose of fueling the ATP synthase. All right, then, let's look at sort of the full story here, the full snapshot of how this all goes about. Here's a really complicated diagram that I'm going to walk you through. It's more complex than you have to know, and I'll try and guide you through the important bits of it. But in essence, the way that we go from a molecule like glucose to stripping it all apart to making energy at the ATP synthase, there's three main steps along that journey. The first is what's called glycolysis. And glycolysis, the word literally means lysis means break down, glyco means sugar, so it's the process by which sugar is broken down. The next step is called the citric acid cycle. And then lastly, we have the final step, which is called oxidative phosphorylation. Okay, in glycolysis, we start off with a molecule of glucose. This is your sugar molecule, and it's a 6-carbon molecule. Now, if you were taking a biochemistry class, you'd have to know all of these intermediate steps, and you'd also have to probably be able to draw them all. But in this class, you just have to know you start off with glucose. It's got 6 carbons, you end up with 2 3-carbon molecules. So the math there checks out. Start off with 6, end up with 2 that are 3 carbons. And those 3-carbon molecules are called pyruvate. There is an energy investment part to this pathway. So 2 ATP are spent here to get it halfway through, and then from there it unfurls, and 4 ATP are made. So the net gain here is 2 ATP produced, even just here in glycolysis. Glycolysis happens in the cytoplasm, so it's not in any particular organelle. And it's a really quick way to make energy. It's a really inefficient way to make energy, but it happens really fast. And it doesn't require oxygen. So like some of your muscle fibers are more suited to make energy through glycolysis, and they have, we call them glycolytic fibers. They're the ones that are, if you're eating a turkey dinner, those are the ones that are more of the white meat, the bulkier muscles, those that need to make a lot of energy quickly. We call them fast twitch fibers. It's all beyond the scope of this class, but some cells, like for example in your body, they only make energy this way, like red blood cells. Red blood cells are basically big old bags of hemoglobin that carry oxygen. They don't have really many organelles in them. They're not spending a lot of energy doing many things. They just mostly are like passive taxis that hold on to oxygen. So they don't have mitochondria. They just make energy through glycolysis. But it's highly inefficient. If we want to go the extra mile and carry on to the citric acid cycle so that we can carry on to the ATP synthase and operate that machine, then we're going to get some serious bang for our buck. Okay, so from here, pyruvate is going to pass through the membranes of the mitochondria, and this is sort of an intermediate step. We call it the oxidation of pyruvate. It becomes acetyl-CoA, and acetyl-CoA gets fed into the citric acid cycle. Citric acid cycle is also sometimes called the Krebs cycle, after the name, I believe, of the scientist who discovered it. Okay, the cycle is called a cycle because it just is a series of biological pathways that continually churn about. Okay, this thing just goes and goes and goes. Citrate turns into ketoglutarate, which turns into succinate, which turns into fumarate, which turns into oxaloacetate, which turns back into citrate. It just keeps going back and back and around and around. But the thing that fuels it or drives it is the constant introduction of pyruvate into the cycle. So as pyruvate enters, it churns the cycle. Now, the big things that are made here, pyruvate will be completely dismantled and stripped apart in this biochemical cycle. And what happens is the electrons and the hydrogens are gathered, and then the CO2 is going to be released as a waste product. So this is where a lot of our CO2 is made, right here in the inner matrix of the mitochondria. And that's a byproduct. We don't use it for anything. You breathe it out, and if you don't breathe it out, you will become acidotic and die. Okay, so it is very important that you do breathe out that CO2, and that's why we breathe. Okay, but one of the things that's very valuable. Well, let me back up. This is a byproduct to us, but to plants, it is the nutrient of life. So that's sort of a foreshadowing to our next chapter here, the really cool symbiotic relationship we have with plants. Our waste becomes their life. I think it's really cool. Okay, but going back, what's really made here is that's of use to us is these molecules here. This is NADH, stands for nicotinamide I can't remember what it stands for, but it's NADH. Think of it as being an electron and hydrogen shuttle. Okay, so you start off with this precursor molecule here, NAD plus, and 2 electrons and 2 hydrogen ions get loaded up onto it. So when you ask yourself where those hydrogens went that was originally in the glucose, this is the answer here. They get loaded up onto these transports, NAD and FAD. The nuance between NADH and FADH2 we're not going to pay attention to. Just know that NAD and FAD are carrying electrons and they're carrying hydrogens. They're each carrying, if you see it as NADH, it's the full version of NAD+. if you think about the math here, you have a molecule that's got a positive charge. So if you add one electron to it, it neutralizes that charge, and then add to it a hydrogen atom, that's another electron and another hydrogen. And so then here you have another pretty sophisticated cellular process here. This is what's known as the electron transport chain, and it is a component of oxidative phosphorylation. And this is where electrons are going to be gathered from NAD and FAD. They're dispensed at different places. And as the electrons whirl across these series of complexes, the electrical attraction they exert on hydrogen ion pulls hydrogens into this space up their concentration gradient. Anytime you bring something up its concentration gradient, remember, to push something up its concentration gradient means to go from low to high. Okay, so these are accumulating. This is becoming the proton gradient, the fuel for the ATP synthase. And it takes energy. For the more hydrogen ions that are accumulating here, the greater energy it takes to push another one in there. And so that energy is provided literally by the electrical attraction that hydrogen ions have, protons have to electrons. The electrons are passing through, being shuttled across these complexes. Hydrogen ions, the protons, they want to attach to them, but they cannot for whatever reason. I don't really know the biophysics involved there. And so they are then pushed out and sort of tricked in a way out to the intermembrane space where they accumulate. And then once they accumulate, then they can flow down through the water wheel. They can flow down through the rotor of the ATP synthase and be used to make ATP. Here you have an adenosine diphosphate and an inorganic molecule of phosphate. The two combine together and they form ATP. Now, what happens to some of these then ultimately is once the electrons have passed through the electron transport chain, they have to get loaded up somewhere. Electrons have to be handled in a very cautious manner. They can't just be bebopping in the cell. Electrons sort of need like a chaperone. They're like middle schoolers. You can't just let them run around in the school. Okay, they gotta have a chaperone with them. Okay, so the electrons, they get loaded up onto oxygen. Oxygen is what becomes known as the final electron acceptor. And this is sort of the other side of the coin here of why we breathe. We breathe not only to get rid of CO2, a waste for us, but also to bring in oxygen. And this is what it's used for. It's used to take those electrons coming off the electron transport chain. Without them, if there wasn't sort of a bunch of oxygens dissolved here in solution ready to take electrons, these electrons would come off of the final complex there, the ETC, and now they would start rattling around in your cells, causing biochemical havoc, right? An unchaperoned electron is called a free radical, and you've probably heard that term before, and they can cause biochemical damage. They can run into molecules, cause mutations, disrupt cellular processes. They're not good. So this is why it's important to always have some oxygen on hand when we're using oxidative phosphorylation. So all the oxygen you breathe, here's where it ends up. It ends up in the inner matrix of your mitochondria so that it can take electrons coming off the electron transport chain. Also, any of the hydrogen ions that have flown through the ATP synthase, if they're accumulating here, they combine with the oxygen. So the oxygen in combination with the electrons, in combination with the hydrogen ions that have flown through the ATP synthase, they combine together to form this really beautiful byproduct that is not harmful to you at all, water. So, pretty cool situation there. Now, this is called oxidative phosphorylation because an important or essential part of it is that oxygen is, wait, let me back up. An important part of it is that it requires the loss of electrons. It requires the oxidation of NADH. Oxidation means any time a molecule is losing its electrons. And so you have all of these NADs and FADs showing up on the scene losing their electrons. and it's the loss of them that drives the whole process. And it's called phosphorylation because we're adding a phosphate group onto an ADP. In other words, we are phosphorylating the adenosine diphosphate. So oxidative phosphorylation. Highly efficient. You will use this sophisticated cellular machine. If you have oxygen on hand, your cells will preferentially use this really sophisticated cellular machine to phosphorylate ADP to make ATP. Okay, in your study guide you have this figure, and you want to know this figure inside and out. Much of the questions from this chapter are going to be just directly off of this figure. So you want to be able to know, okay, what is this molecule that I'm starting with? What is this molecule I'm ending up with? What is this process called? What is this process called? And what are these products that I'm making here? You want to know, what are these things that are accumulating here? You want to know, what is this little cellular device? What is this cellular device? And what are some of these products that are made when the electrons come down? What do they bind to? And, in combination with some of these little pink dots, what is the product made here? And then lastly, what's the golden ticket? What is the main event? What is all of this about? What are we producing here? What's the end of the story? Okay. You want to know what all of those labels are. The letters represent the processes, so this would be glycolysis, citric acid cycle, or the Krebs cycle, oxidative phosphorylation. And then all the numbers just represent what are the names of those things that we're looking at. Okay, work your way in and out of this diagram. Get a nice cup of coffee or a tea. Quiz your peers on this diagram. A good portion of points on the exam from this chapter will come just right off this figure. This very figure will be on the exam. Okay, now there's a couple other things that can happen that we won't get into, like fermentation. If oxygen's not around, then we have to find some way to keep glycolysis going. Sort of the regeneration of NAD. If oxygen's around or not around, it leads to different products. If oxygen's around, it leads. If oxygen's not around, it starts to lead to lactic acid, and then we have to find a way to regenerate, and so then there's fermentation and stuff like that. But we won't go into any of that detail here in this class. If you wanted to learn about it, you certainly could. It's related to what we're talking about here if you're curious to know what's happening in the process of fermentation. But I'll leave you here with just another look at the ATP synthase, and just take a moment to think about how beautifully made you are. Think about how you are a collection of trillions of cells. Those cells are doing an incredible amount of things to stay alive. And one of the main power sources, the generator that fuels all of those products. Think about entire genomes, billions of bases being replicated, and sugars being, macromolecules being made, and pumps operating, all that activity being driven from this really incredible cellular machine. When Albert Einstein says, the more I study science, the more I believe in God, you start looking at things like this, and you start to think to yourself, I don't know if this is all just a curious accident, right? There's a lot of ingenuity here. And my take on it is that it all comes from a really brilliant mind. And I hope you're taking one more step into my goal for you in this class of just having more reverence for the creative mind that's behind it all, and just letting it be a thing that helps you to trust in Him, trust in God, knowing that the one who could design something as elaborate as this can surely take care of your needs and wants and your desires. So trust Him, and He is able. He is powerful. He is worthy of our praise, and we can appreciate that even in something like biology.